Welcome to The Explainer. Today we're cracking open the ultimate black box, the human brain. We already know it's a 3-pound marvel sitting right inside our skulls, but how exactly do we know what we know about it? Well, our goal today is to dig into the brilliant, incredibly precise, and honestly sometimes mind-bending methods researchers use to decode the mind. We're going to open up the neuroscientist toolkit and see exactly what these incredible tools teach us about the brain. Get ready, because we are diving deep into how we look inside the mind.
Here's our roadmap for today's explainer. We've got 6 sections. First, the foundation. Then, Tool 1, subtraction. Tool 2, visualization. Tool 3, observation. Tool 4, manipulation. And finally, the synthesis. Okay, let's dive right into this. Section 1, the foundation, the core strategies of neuroscience. Before we pick up a single tool, we really need to set up the overarching framework that anchors absolutely all neuroscience research.
Every single experiment starts with a strategy, right? And research methods in behavioral neuroscience are basically structured around 3 primary ones. First up is somatic intervention. This is where researchers actually manipulate the body's structure or chemistry to see how behavior changes. Then there's behavioral intervention, which kind of flips that on its head. We alter the behavior or the environment and then observe the resulting physiological or anatomical changes in the brain. And finally, correlational studies. These just measure the extent to which a somatic variable and a behavioral variable co-vary. Grasping these 3 strategies is absolutely foundational before we pull any specific tools out of the bag. Moving on to Section 2. Tool 1, subtraction, lesions and brain mapping. Let's transition to the oldest method in the neuroscientist toolkit. Think about it. To figure out how a complex machine works, sometimes you just have to remove parts to see what breaks. We call this subtraction. When researchers use experimental ablation, or lesion studies, they're essentially destroying or inactivating a portion of the brain and evaluating the behavior that follows. This lets them infer what that specific region actually does. And they've got a few options here. They can use radiofrequency, or RF lesions, which use an alternating current to generate heat. This completely destroys all nearby tissue. We're talking both cell bodies and axons. But if they need more precision, they use excitotoxic lesions. These use excitatory amino acids to basically stimulate neurons to death, but crucially, and this is a big deal, they spare the axons that are just passing through the region. We can also do temporary lesions. Using anesthetics or a GABA stimulant called macemol, researchers can literally turn off a region just long enough for the animal to serve as its own control. But to ensure good science, researchers heavily rely on a critical control, the sham lesion. This is where they perform all the surgical steps, the anesthesia, the incision, lowering the electrode, but they skip the actual tissue destruction. This is absolutely crucial to make sure any behavioral deficit is caused by the missing brain region itself and not just the physical trauma of the surgery. But you might be wondering, how do you actually reach a tiny structure deep inside the brain without destroying literally everything on top of it? Well, the answer is stereotaxic surgery. Researchers use a stereotaxic apparatus. It holds the head in a fixed position and allows an arm to move an electrode or pannula through measured distances in 3 spatial axes. They pair this with a stereotaxic atlas, which is basically a 3D map of the brain, and they navigate using a physical reference point on the skull called the brygma. It's like a highly precise microscopic GPS system. Section 3. Tool 2. Visualization, histology, and tracing. Okay, so you've successfully targeted a region and tested a behavior. But in science, you have to verify your work. You have to literally see where your lesion actually ended up. And that brings us to Tool 2. To actually see these structures, scientists rely on a 3-step histological method to prepare the brain tissue. Step 1 is fixation. You've got to preserve the tissue with a fixative like formalin to stop autolysis and decomposition. Step 2 is sectioning. Here, you slice the brain into incredibly thin sections using a machine called a microtome or a cryostat if it's frozen. Step 3 is staining. Since brain tissue is mostly transparent, we use stains to actually see anything. Nissl stains use basic dyes to highlight RNA and cell bodies, which is just perfect for studying cytoarchitecture and counting cells. Golgi stains, on the other hand, soak the tissue in silver chromate. This is wild because it randomly stains only a tiny percentage of neurons, but it stains them in their entirety, revealing these absolutely beautiful, fine structural details like branching dendrites. Beyond just looking at static cells, we need to map the brain's wiring diagram to see how regions connect. And this brilliantly illustrates the concept of tracing. Anterograde labeling traces efferent axons. It moves forward, starting from the originating cell body out to the terminal buttons. Retrograde labeling does the exact opposite. It traces afferent axons moving backward, from the terminal buttons back up to the originating cell body, usually using chemicals like horseradish peroxidase. These tracing methods teach us exactly how the neural highways are laid out. Now let's see how this builds into some mind-bending modern technology. We aren't limited to just looking at thin, dead slices anymore. With a method called clarity, scientists replace light-absorbing lipids with a water-soluble gel. This renders the brain literally transparent. It allows for 3D visualization of deep structures without any physical sectioning. It's wild. Then there's Brainbow, which uses genetic manipulation to make neurons express hundreds of different glowing fluorescent hues, completely disentangling complex connections. And hey, if you want to see a whole chain of neurons, we use transneuronal tracing. This employs weakened viruses, like rabies actually, that jump across synapses to map out entirely interconnected neural networks.
Section 4. Tool 3. Observation. Imaging the living brain. Up to this point, we've mostly been talking about animal models and postmortem tissue. But obviously, we need to apply these concepts to humans, so we need Tool 3.
I want to pose a question directly to you. How exactly do we study a living, thinking human brain that we cannot physically touch?
The answer, of course, lies in non-invasive imaging, and it's super vital to divide this into two distinct categories: structural versus functional. Structural imaging takes a static picture of the brain's anatomy. We're talking about things like CT scans, which use X-rays, MRI, which uses strong magnetic fields for a much higher resolution, and DTI, which tracks water diffusion along axons to visualize white matter tracks. Functional imaging, however, is completely different. It's like recording a video of the brain thinking. It actually measures metabolic or magnetic activity in real time.
Let's look a little closer at those functional techniques. A PET scan measures regional metabolic activity by having the patient ingest a radioactive tracer, like 2-deoxyglucose. Then we have fMRI, which is probably the most famous one. It tracks the BOLD signal, that stands for blood oxygen level dependent signal. This acts as a proxy for neural activity by measuring the ratio of oxygenated to deoxygenated blood. Finally, we have MEG. This uses incredibly sensitive devices called squids to detect the minute magnetic fields produced by synchronously active neurons. So the crucial point here is that while fMRI gives great spatial resolution, showing us where things happen, MEG offers much higher temporal resolution, showing us exactly when they happen. Section 5. Tool 4. Manipulation, recording and stimulating activity. Okay, so we've passively watched the brain. Now it's time to actively listen to it, and even hijack its electrical signals. Welcome to Tool 4.
To study electrical signals, researchers use different scales of recording. Think of microelectrodes like a microphone held right up to a single instrument in an orchestra. They record the activity of single neurons. Macroelectrodes, on the other hand, specifically an EEG attached to the scalp, are like recording the entire orchestra from the back of the concert hall. They record the summed electrical activity of millions of cortical neurons at once. And to make sense of all that chaotic noise during a specific task, researchers use event-related potentials, or ERPs. This involves averaging dozens of EEG trials together to isolate the specific electrical signature of a single cognitive task.
But here's the thing. We don't just record. We can stimulate. And honestly, nothing in neuroscience right now is as wildly fascinating as optogenetics. This method employs genetically modified viruses to insert light-sensitive ion channels into specific neurons. Once they're in there, scientists literally use colored light to control the brain. Shining a blue light causes depolarization, which excites the neuron. Shining a yellow light causes hyperpolarization, which inhibits it. We can turn specific cells on and off with a literal light switch. It is absolutely remarkable.
Section 6. The synthesis. Neurochemistry and robust science. Well, we're reaching the end of our explainer today, and it's time to bring all these fantastic tools together.
The final pieces of our toolkit teach us about neurochemistry. If we want to know what chemicals are floating around in the brain, we use microdialysis to collect extracellular fluid. If we're hunting for specific proteins, we use immunocytochemistry with labeled antibodies. To see exactly where a specific gene is actively turned on, we rely on in situ hybridization. And to identify exactly which neurons were active immediately before our analysis even began, we measure the FOS protein.
We have covered a massive amount of ground today, exploring this toolkit, from heat lesions to glowing genetic brains, all the way to magnetic scanning. But if there's one thing you take away from this explainer, let it be this quote directly from our source material. Because different procedures can produce contradictory results, a comprehensive research program often compares findings across these various methods to reach robust conclusions. Look, no single tool is perfect. Real, robust science requires layering, subtraction, visualization, observation, and manipulation all together.
The brain is the most complex structure in the known universe, and frankly, we are just beginning to understand how to read its wiring. Now that you understand the toolkit, and now that you know exactly how scientists do their work, the real question is, what mysteries will you uncover when you open the black box of the brain? Keep exploring, keep learning, and we'll catch you in the next explainer.